Hello again. We are still in chapter 2, which is on cell structure and function. Okay, so after I have explained to you on subtopic 2.1, so now we are going to proceed on subtopic 2.2, which is on structure and functions of cell membrane and organelles. Okay, so under 2.2, there are several learning outcomes that we have to achieve. So the first one, I will show you the detailed structures of typical plant and animal cells, and also I will state the organelles that are present. And the next learning outcome that we have to achieve for this subtopic is to be able to explain the structures and functions of the organelles that are present in eukaryotic cells. Okay, so there are two types of eukaryotic cells, which are animal cell and plant cell. These two types of cells are differentiated mainly based on its structures. In this slide, it shows the structure of animal cell under both electron and also light microscope. So as you can see, in general, the animal cell is much more complex compared to the prokaryotic cell, which I have explained to you in the previous videos. The animal cells that you can observe here contains mainly plasma membrane, cytoplasm, and nucleus. And this is a diagram of an animal cell under a light microscope. While for this slide, it shows the structure of a plant cell under both electron microscope and also light microscope. And in plant cells, structures that can be clearly seen under electron microscope are cell wall. So this is the one that differs plant cell with an animal cell. And another structure that can be seen under electron microscope are plasma membrane, cytoplasm, nucleus, vacuole, and also chloroplast. But under light microscope, you can only see few organelles such as chloroplast and also cell wall only. Okay, so next I am going to explain the structures and functions of organelles such as nucleus, rough endoplasmic reticulum, Golgi body, lysosome, ribosomes, mitochondria, chloroplast, and also centriole. Okay, so for the first organelles that I am going to explain is on nucleus. Okay, so nucleus is the largest organelle and it is spherical in shape, and the inner part of the nucleus contains nucleoplasm. So as you can see, it is spherical in shape, and also you can see that the nucleus are the largest organelle that contains in the animal cell. The nucleus are enclosed by two layers of nuclear membrane. So as you can see here, there's a two layer of nuclear membrane, and the nuclear membrane fuses to form nuclear pore, this part here, okay, which functions to regulate the movement of molecules across nuclear membrane. The nucleus also, this nucleus also contains nucleoplasm. Nucleoplasm, which is a jelly-like fluid which contains nucleolus and chromatin. So what is nucleolus? So nucleolus, this part here, okay, this part here is a spherical shape also, but it is non-membranous. Compared to the nucleus, it's membranous, double membrane. But this nucleolus is non-membranous and doesn't have membrane, and it contains DNA, RNA, and protein because it is a site of rRNA synthesis. While chromatin is a long protein, chromatin is a long thin strand of DNA and also histone protein. So here is the nucleus, and here is the long thin strands of DNA and also histone protein. So the nucleus basically functions to control cell activities and cell division, store genetic material, site of RNA synthesis, and also for the assembly of ribosome subunits. Okay, so the next organelles that we are going to discuss is on endoplasmic reticulum, ER. Okay, so this is a structure of an ER, endoplasmic reticulum, and as you can see, it is an extension of nuclear membrane. So this endoplasmic reticulum is actually an extension of the membrane of the nucleus. Okay, so this endoplasmic reticulum consists of two types, which is rough endoplasmic reticulum, and also smooth endoplasmic reticulum. Okay, so the rough endoplasmic reticulum are flattened sacs. Okay, so as you can see, the sacs are flat. Okay, and it has the dotted-like structure, which is ribosomes that are attached on the outer surface. So this rough ER appears granular. Okay, and while for the smooth ER, SER, okay, it consists of a more tuber-like sacs. So as you can see, the sacs are in the form of tubes. Okay, and they lack of ribosome, and as you can see in the picture also, there are no dotted-like structures. So it means that it doesn't have any ribosomes on its outer surface, so it appears smooth. So that is why it is called smooth ER. So both of the endoplasmic reticulums have different functions. So for the first one, the rough endoplasmic reticulum, RER, function for intracellular transport of protein, which means it will package and transport protein to Golgi body within a transport vesicle. So as you can see here, so this is transportation of proteins. Okay, so from endoplasmic reticulum to the Golgi body. Okay, so the next endoplasmic reticulum is the smooth endoplasmic reticulum, and it functions as a site of lipid synthesis, detoxification of toxic waste, for example, drugs, poisons, and also the storage of calcium ions, specifically in the skeletal muscle. Okay. Okay, so the next organelles that we are going to discuss is on Golgi body. Okay, so Golgi body. So Golgi body, here is the structure, basic structure of this Golgi body. And as you can see, it is enclosed by a single membrane. Okay, a single membrane consists of stacked, flattened membranous sacs that are not physically connected, called cisternae. So this is the cisternae. Okay, so this is the cisternae. Here, one cisternae, and this is another cisternae. Okay, and the space within the cisternae is called a lumen. So remember, space is the lumen. Space is the lumen. Okay, so each Golgi stack has cis phase and trans phase. Okay, so the cis phase is facing towards the nucleus. So as you can see, here is another diagram. So I will show you based on this diagram. So cis phase is facing the nucleus or the rough ER. So this is the rough ER. And while the trans phase is facing the plasma membrane of the cell. So here is the cell. So trans phase is facing outwards. Alright, so for this slide, I am going to explain on the mechanism that occurs in Golgi body. Okay, so it starts with endoplasmic reticulum, ER, where it pinches off, as you can see here. So this is the endoplasmic reticulum. It will pinch off to form transport vesicle. So here is the transport vesicle, which move towards the Golgi body. Okay, so here is the Golgi body and fuses with the cis phase of the Golgi body. So remember, the cis phase is the one that is facing the nucleus. Okay, and releases its content such as proteins, carbohydrates, lipids into the lumen of the Golgi body, and then the contents will be then modified as it moves from the cis phase to the trans phase. Remember, the trans phase is the one that is facing the plasma membrane of the cell. So the tip of the trans phase will then pinches off to form secretory vesicles. So here is the secretory vesicles which move towards plasma membrane and fuses with it. So it moves towards plasma membrane and fuses with it to release its substances by exocytosis, or it will also remain in the cell as lysosome. So basically, Golgi body have two functions, which is the first one is to process, modify, sorting, package, and also transport protein. And then the second one, the second function of Golgi body is to form lysosome. So here is another diagram where you can see it starts from the ER. Okay, it forms the transport vesicles, which fuses to the cis phase of the Golgi body, where the Golgi will then do the processes, the 5 processes that occurs, and will then be pinched off from trans phase, and will then either fuse with the plasma membrane and release its substances outside, or stay in the cell and form lysosome. Okay, so for the fourth organelle is lysosome. So as you can see here, this is the structure of a lysosome. It is enclosed, it is spherical in shape, sorry, so it is spherical in shape. Okay, it is enclosed by a special single membrane which may contain hydrolytic or digestive enzyme. So inside here got enzyme. Okay, so basically the lysosome got three functions. Okay, so the first one is as an intracellular digestion, this one. Okay, so intracellular digestion is where the lysosome will digest macromolecules which enter the cell by phagocytosis. So in this diagram, as you can see, there's a substance from outside of the cell which enters the cell, and this substance will then be digested by the hydrolytic enzyme that contains in the lysosome. And second function of lysosome is to recycle the cell's organic matter, where the lysosome, so this is the second function. Okay, so where the lysosome will digest old and damaged organelles. So in this diagram, as you can see, this lysosome will digest these old and damaged organelles such as mitochondria or chloroplast by autophagy process. Okay, so the hydrolytic enzyme inside the lysosome will be released, and will digest the organelles. And lastly is programmed cell destruction. Okay, so this is where the lysosome digests the whole cell, okay, by autolysis. So this is where the lysosome automatically releases the enzyme inside it. So when the lysosome releases the enzyme, so the whole cell will be affected. Okay, so for example, during metamorphosis and also during the development of human embryo. Okay, so in this slide, I am going to explain about endomembrane system. Okay, so this endomembrane system. So what is endomembrane system? Endomembrane system includes nuclear membrane. So this is the nuclear membrane, endoplasmic reticulum. This is endoplasmic reticulum. Golgi body. So here is the Golgi body. Lysosome. Okay, lysosome. And also vesicles or vacuole, and also the plasma membrane of the cell. So endomembrane system is a group of membranes. So basically it means that a group of membranes and organelles in eukaryotic cells that works together to modify, package, and also transport lipids and proteins out of the cell. Okay, but this endomembrane system does not include organelles such as what? Such as mitochondria and chloroplast. So here is another diagram that shows the endomembrane system. So as you can see here, this is the nucleus. So it is connected with the rough ER, the smooth ER, and also the Golgi body. And lastly is the plasma membrane, which not forgetting also the vacuoles, vesicles, and also lysosome. Okay, so for the fifth organelles that we are going to discuss is on ribosomes. Okay, so what is ribosome? Ribosome is a small granule, spherical in shape, non-membranous, and made up of rRNA and proteins. And ribosome mainly consists of two subunits, as you can see here, large subunit and also a small subunit. Okay, so there are two types of ribosomes, a free ribosome and also a bounded ribosome. So basically the free ribosome lies freely in the cytoplasm and synthesizes proteins that function in the cytosol. Okay, while for the bounded ribosomes, the ribosomes are attached to nuclear membrane of rough ER that synthesizes proteins for export outside of the cell or in the lysosome. So basically, ribosome has two functions, which is the site of protein synthesis, and also it acts as enzyme to catalyze the formation of peptide bonds. Okay, so in this slide, it shows the 6 organelles that we are going to learn, which is mitochondria. Okay, so mitochondria here is the basic structure of mitochondria. So as you can see, it is oblong in shape and also a cylindrical in shape also. It is enclosed by 2 layers of membrane, so which consists of outer membrane and also inner membrane. So the outer membrane is smooth, as you can see here, is very smooth. Okay, means it is not folded and it doesn't have any ribosome attached to it. Okay, and it points outwards towards the cytoplasm. So you just imagine this part here, outside here, is the cytoplasm. Okay, so while the inner membrane is folded to form cristae. So this is the inner membrane and it folds up to form a cristae. Here is the cristae. So the space between the outer and the inner membrane is called intermembrane space. So in this diagram, the intermembrane space is this part here. Okay, so the inner membrane encloses a fluid-filled space called matrix. So this blue area is the matrix, which contains bacteria like DNA and ribosomes to synthesize its own protein and enzyme that are involved mainly in Krebs cycle. Okay, so for the 7th organelle, which is the chloroplast. So here there's a diagram of chloroplast. Okay, so as you can see, the chloroplast is shaped is oblong and also is biconcave or biconcave shape. It is enclosed by 2 layers of membrane also, similar like the mitochondria. Okay, there's outer and also there's an inner membrane. The outer membrane also same as the mitochondria, points outwards towards the cytoplasm, while the inner membrane, so this part is the inner membrane. Okay, inner membrane encloses a fluid-filled space called stroma. Okay, so what is stroma? Stroma is here. So stroma contains bacteria like DNA and ribosome to synthesize its own protein and enzyme. Same or similar like the mitochondria, it has its own DNA and ribosome that are involved in Calvin cycle, which differs with the mitochondria, it involves in Krebs cycle. The space between outer and inner membrane, outer and inner membrane space is called intermembrane space. Okay, the embedded within stroma are membranous system called thylakoids. So this is a thylakoid. So this is a system called thylakoids. So what is thylakoids? So it is a flat disc which are stacked to form grana. Okay, so what is grana? Grana are interconnected by intergrana membrane, or we can say as lamella. As you can see here is the thylakoid system. Okay, it is a stacked sacs. So the grana are interconnected by a lamella structure called lamella. So the chlorophyll and photosynthetic pigments are embedded within thylakoid membrane. So the chlorophyll contains inside the thylakoid membrane here. So after we have observed the structure of the chloroplast, we now have to see the function of the chloroplast. So chloroplast mainly function as a site of photosynthesis. So where the light dependent reaction occurs, which is in grana, and also the light independent reaction occurs, which is in stroma. Okay, and also chloroplast will store the food of the plant in the form of starch. Okay, which is stored in stroma. Okay, so if you realize in the previous slides where we discussed about mitochondria and chloroplast, there are some similarities between the two organelles. Okay, so one of the similarities between the two organelles are they are semi-autonomous organelles, which means both of them, okay, can duplicate and reproduce independently within the cell. Okay, which means it doesn't it has, because it has its own DNA, it can produce its own protein, so that's why it can reproduce independently. Okay, so for the last organelle, which is number 8, which is the centriole. Okay, so centriole is located in a region called centrosome. So here is a cell. So this part here is called the centrosome. So that is the region where centriole is located, near nucleus of an animal cell. So it exists in pairs. So as you can see here, in pairs, and is oriented at 90 degrees angle to one another. Okay, so as you can see here, 90 degrees to another. So each centriole is composed of 9 sets of triplets microtubules. So 9 sets of triplets microtubules. So this, as you can see, this is a one pair of the centriole. So this is when you observe cross section of the centriole. So it says it composed of 9 sets of triplets microtubules. So there's triplets here. So there's 9 sets of it. Alright. Okay. Arranged in a circle. If you still remember, in my previous video, we discussed about 9 plus 2 microtubules. So that is more to the flagella structure. So this one is centriole. It consists of 9 sets of triplets microtubules. So basically, the centriole functions to organize microtubules during the cell division. So for the next topic or the next chapter, which is in mitosis or meiosis, you will learn about cell division, and you will also learn about this organelle, which is centriole.